I'm going to give a talk today that's a little bit of, sorry, I have to learn to stand still. I'm going to give a talk today that's a little bit of the history, a little bit of Singapore in terms of longevity, a little bit of science, a little bit of religion in terms of what's happening in the longevity field and the belief that we can actually make people live healthy longer. And so it's a bit of a combined talk. But I wanted to start with this. I got asked to do this for a recent conference, so I thought I'd just include a few slides. And you know, the longevity field has grown like crazy in the last 10 years. It's probably the fastest growing sector of the biotech area. It's their longevity clinics, and I'll talk just briefly about those. But this is a long process, and basic research has really set the stage for where we are in terms of understanding aging. And in fact, actuarial tables began the whole question back in 1825 when Gompertz came up with a mathematical law of human mortality. And it's amazing the durability of that law because, you know, in the recent gerophysics conference that we had, that was really still a starting point for trying to write mathematical equations that explain what's happening during the aging process. The first intervention that slowed aging was around 1935 with calorie restriction. The first really mature evolutionary theories of aging were in the 1950s with Medawar and Williams, and they're still sort of the basis for thinking about how aging happens from an evolutionary perspective. And then things started to accelerate. We had genetic mutations in the 70s and 80s, cloned the genes in the 90s and the 2000s. The first drug that really, I think, had a major impact on the field was rapamycin in the late 2000s, extending lifespan, actually crossed the 2000s in yeast, worms, flies, and mice. Showing conserved pathways modulating aging was around the late 2000s, 2009 or so, the first quantitative study. And that's, when you think about it, everybody accepts that now, but it's really fascinating that everything from a single-celled yeast to a human have similar genes and pathways that modulate the aging process. I certainly didn't expect that when I started working in yeast a long time ago. And then aging clocks in the last decade or so, and the hallmarks and pillars of aging papers, which I think are transformational because they help the rest of the world think about aging. And I'll talk about that for a little bit. And I have to give credit where credit's due. This was really driven in large part by the National Institute of Aging in the US. It was the first institute focusing on aging, and you can look at the money they've spent over the years in the United States on the basic biology of aging. And this is really why most of the discoveries have come from the US since that period. The resources were there to do the experiments. And still, most other funding agencies are not heavily focused on the aging process, although Singapore's starting to change that, I think. There's also been a number of philanthropic organizations that have really supported this field. I've just listed some. There are many others. These are some of the big ones, and I'll just point to Hevolution, which is funded a nonprofit foundation by the Crown Prince of Saudi Arabia, which has already spent $450 million on longevity research. And they got into a kind of a pause mode, but I still think their plan is to really start funding grants big and small in Asia within the next year to two. So that should be grants available to everybody. And a lot of the research was from preclinical models. People ask me why we don't, talk about, psychology and anxiety and things like that in terms of the aging process when they clearly are heavily involved. And I think a lot of it came from the development of the pathways that govern aging using mostly the preclinical animal models. So a lot of major discoveries in yeast, worms, and flies, and also rodents. A lot of early cell culture work. Epidemiology in humans has been critical to feeding this understanding, and also heavy focus on disease of aging. But where that's going now, and I want to point out that I'm going to talk mostly about humans today, but I think that there's still a dramatic need for the basic sciences. But we have to use the preclinical models to ask the right questions. And so these are some of the questions I think are are really critical. So first of all, it's really hard to run a yeast lab or a worm lab these days. It's just very difficult to get funding anywhere for that. But there's still key questions, mainly around systems biology and network biology and the aging process that can really only be answered in these models. And so they still need to be supported. A lot of transition from cell culture to stem cell culture work. Human data is being analyzed. First of all, there's a lot more human data. And second of all, it's being analyzed in much more progressive or creative ways now with the AI revolution that's happening. Better design of animal longevity studies. I think a lot of people now are thinking about how we design a mouse aging experiment to better teach us about humans. And also, as you heard this morning, with organoid culture, trying to develop human models that better mimic the human process. And also what's happened more recently is the importance of canine studies. By that I mean not lab dogs, but companion animals, and those are a fascinating model for longevity research. And there's an increasing amount of effort in the private sector to make your pets live longer too. So I think that's a very exciting area. Primates are important, but they live a long time and they're very expensive, so they're still not widely used in this field. There's been two sort of major conclusions, from all of this. And this is a slide that I just saw Matt Kaverline a couple days ago in Mauritius, and we realized we've been using some version of this slide for 20 years, which is kind of sad, but I think it's still apropos in that we need to move from sick care to health care. We need to stop focusing on diseases as if they're in isolation, and that's either research or clinical practice. And understand that aging is the biggest driver of all these chronic diseases, many others as well, and also functional decline, and really begin to understand the aging process. That's what a lot of this research has taught us. And also this hallmarks and pillars. These papers are old. There's hallmarks on the left. At that point, there was 9 of them, and now the last paper I saw, there were 24, so they're increasing. And pillars, there were 7. This was from an NIA-based conference in 2013. I don't think it's important to talk about each of these in detail. There are lots of research on all these areas. I think what's more important is to look at the connectivity of these pillars. There are lots of different processes linked to aging, but I really think, based on a number of different reasons, that we're looking at a homeostatic network here that governs the aging process. Your body is trying to keep you healthy as long as possible, and it's responding to the damage, the stochastic events, your bad behaviors, whatever, and trying to make adjustments to keep you healthy. And when that process breaks down, that's when you start getting susceptible to various chronic diseases. And you see that from the interventions that affect longevity. They don't affect just one of these pillars or hallmarks. They affect all of them typically, or most of them. And I think that's because they're hitting nodes in these networks and preserving the function of the network rather than specifically targeting one process. And then if you look at the longevity industry, just a couple slides on this, it's growing like crazy. There are biotech companies in the space, and I'll talk about that a little bit. Supplement and nutrition companies. Longevity clinics are happening everywhere now, and I can't even keep track of how many of them there are. Diagnostic companies, biologic aging clock companies, for instance, and consumer companies are very interested in how to incorporate longevity into their business sector. All of these are interesting areas, and I think this is a big enough problem that we need cooperation between academic and private sector to really make this go as fast as possible and increase human health span. This is hard to see. It's okay. I think you can see it well enough. I stole this slide from Phil Newman at Longevity Technology. And this is the amount of spending in the longevity sector by year. And you can see it's going up. There was a bit of a blip in 2023, but it's going up dramatically as we go forward, which is a positive sign. I don't want people to get too excited, though, because if you look at that red line on there, that's spending on longevity research compared to all the other lines. And the one that I want to point out is oncology, which is in blue, which is still 5 or 6 times higher than what we spend on aging and longevity research. Now, I think if aliens came to Earth and they said, wait a minute, 30% of the population gets cancer and 100% of the population gets aging, and yet we spend 6 times as much on cancer as aging, they would just conclude that there's no signs of intelligent life and leave. But this is obviously for historical reasons. And it's not my intention, by the way, to cut spending on cancer research. I think that's important. But I do think we're still dramatically underspending on aging and longevity, and we need to get more resources to move faster. So this is my version of the slide you heard this morning. But I think it reflects, the numbers are slightly different, but they reflect the same story, which is Japan has longer life expectancy and Singapore has longer healthy life expectancy. But these are, by most measures, the 2 highest countries in the world right now. And that's led to a lot of interest in aging in Singapore. And in fact, you know, Singapore got identified as the 6th blue zone by Dan Buettner a couple of years ago. Okinawa is probably the most famous of those, but there are 4 others around the world. But Singapore was differentiated because it was a manufactured blue zone. In other words, the policy of the government, the society, the environment is creating a situation where people can live a long time. I don't know why BBC chose to have someone eating high-carb noodles as a sign for a blue zone, but anyway, it was reported around the world and generated a lot of interest. And so if you look at Singapore in more detail, there's a lot of challenges. You know, there are about 30% of the population is going to be over the age of 65 in the very near future. And the support ratio is down to 2, so that means 2 working people for every retired person. And so if you put this together, and there's some version of this in every country in the world. I was just in Mauritius, and they're a little bit behind in terms of the demographic change, but they're changing dramatically. Every country in the world is changing right now. 2 billion people over 65 by 2050. So if you look at this, I think it's pretty clear that this is the biggest biomedical challenge of the century. It's not COVID, like everybody thinks. COVID actually just reflected the problems with getting older, the loss of resilience, the inability to recover from viral infections. And we need to do something about keeping people healthy longer. Otherwise, we have all kinds of economic problems and quality of life problems that are expanding. Singapore's approach over the years has been very oriented toward managing older people, keeping them as functional as possible, having more and better care, more hospitals, encouraging people to exercise. You can see a pictographic version of that on the left in The Independent, and a more detailed version in The Lancet that was published recently. And this is part of what the success has been. People have gotten healthier here because of this strategy. But we have to go beyond managing the aging population and trying to prevent the detrimental effects of aging. I think healthy older people are the first country that has healthy older people is really going to have a huge advantage over other countries in the world, because if you combine the wisdom that comes with age with the health that comes with youth, you have a massive innovation base. And Singapore is starting to change. I didn't want to talk about any details of this plan because it's not out yet and I don't know all the details. So I used Google AI to source quotes made by government officials about the Research, Innovation, and Enterprise 2030 plan that's going to come out within the next year. And so this is all what Google found that's available online. But you can see that the language is really exciting. Longevity identified as a key national priority for 2030. Improve the quality of life of Singaporeans as they age. Delay biologic aging and prevent age-related diseases to extend health span. Leveraging AI and data science. And so I think that this is a really innovative set of words, and this gives an opportunity for Singapore to really become a leader in this space if this is followed through on. Now, I just want to give an update on healthy longevity for our guests mostly. This program started in 2020. We have about 40 faculty now working on different aspects of aging and longevity. Several of us, and much of what you'll hear about from me, is on the basic pathways of aging and translational approaches to slow the systemic aging process. A lot of expertise also on neurologic aging, vascular aging, muscle aging, and reproductive aging, which I'm not going to go into as much detail today. There's also a Center for Healthy Longevity that operates through NUHS, and it's much more clinically oriented toward testing interventions in people to see if they slow aging. And recently we became part of the Life Sciences Institute as well, which I won't go into in detail. There are a lot of affiliated institutes as well, and so I think that we're really building a environment around healthy longevity. There's a Reproductive Longevity and Equality Center. The Academy for Healthy Longevity by Andrea Meyer is not only doing research, but doing a great job of training the next generation of longevity researchers and longevity clinicians. Chris Chen has a Memory, Aging, and Cognition Center. There's a Center for Sleep by Michael Chi, and Center for Translational Magnetic Resonance Research that's part of our Healthy Longevity program as well. And we also have a number of large cohort studies. You'll hear about SG70, I think, from Ko Won-pui, and you'll hear from Chris Chen as well tomorrow. So the preclinical research around basic aging is really geared toward molecular studies to understand why aging causes disease and functional decline. So working on Alzheimer's is fine if you want to treat Alzheimer's. What we really want to do, though, is understand why does aging drive the risk for Alzheimer's disease. A lot of translational studies have developed new interventions, and they can be lifestyle, small molecules, and even stem cells and beyond. A lot of effort in the program to measure biologic age. I'll show you a little bit of data today, and a lot of epidemiology going on. From the preclinical side, we have all of these systems working now. We have yeast, worms, and flies, mice, killifish, which is an interesting model to test aging because they age really quickly. And then in humans, we use biomarkers of age to validate interventions and also look for chronic disease prevention. Of course, ultimately, the challenge is implementation. If you find things that work to slow aging, how do you implement them in the population? And that one of the things that keeps that we keep focused on in that regard is that when the interventions we're studying and the biomarkers we're studying are scalable. You know, we're not testing $100,000 stem cell injections that can't be scaled to the population. The goal here is to keep billions of people healthy, not billionaires healthy. They can be part of the billions if they want. But I think that we want to scale this to the entire Singapore population. On the translational side, we're testing a lot of small molecule interventions and also combinations of interventions, which is important, looking at lifestyle. And ultimately, it's about personalization and precision. And there's an interesting biologic aging clock that I've been involved with, but mainly developed by Jan Gruber and Feng Sheng. And Jan will be talking about that tomorrow. So we believe this is an actionable biologic aging clock that can be implemented now to improve health in the population. Now, the problem is, how do you do a clinical approach with regard to aging? You know, still the single best marker of aging in any animal model is survival, how long the animal lives. Yeast and worms and flies, that's about 2 weeks to a month or two. That's pretty easy. Mice, 2.5 years, getting more expensive, but doable. No graduate student wants to give 50-year-olds a drug and see if they live longer in the human population. So we have to have other ways of doing that. One proposed way is to use clinical trials to target single or multiple disease prevention. You might have heard the TAME trial, which has had trouble getting funding in the US, which is a tens of millions of dollar trial on 3,000 people to try to prevent chronic disease in 60-year-old population over a 3-year window. That metformin is the drug there, and that costs 4 cents a day, so you can understand how hard it is to fund a project like that. And even if you do, that's testing only one intervention. There are hundreds and thousands of interventions that we would like to test ultimately in longevity. The There's also the biotech approach, and I'll tell you a little bit about one of our projects that fits into this realm, and this is kind of an unfortunate but necessary path you have to take. So if you hit an aging pathway and you want to raise money in the biotech sector, you need a disease indication to try to treat. You need to be able to get reimbursed. And most biotech company, most investors in pharma, for the most part, don't believe aging is an indication yet. And so you have to pick a disease that pathway is linked to go after, and I'll talk about one of those. A lot of what we really want to focus on is clinical trials to target surrogate endpoints, in other words, using biologic aging clocks or some other measure of aging and trying to slow or reverse the progression of those markers. There's also a lot of existing suboptimal data, and I'll just touch on that, and Camille in my lab will tell you a little bit more about that later. And also longevity clinics. These clinics, what we're really encouraging them to do is share data with us or with someone in the field that can analyze the data, because they're collecting data on a wide array of interventions. I can't test in an academic setting, but they may work. And so I think if we could get a data repository from clinics and we begin to evaluate that data, it's not pristine clinical double-blinded controlled data, but it's still very interesting. Okay, so just briefly, I'll tell you a project related to the biotech approach. And so we've been doing a lot of interventions in mice. We don't do survival anymore for a couple reasons. One is it takes forever, and we want to test a lot of different interventions. And another is that we want to harvest the mice at the end of the experiment rather than finding them dead in the cage the next day when you can't do anything. Because if we harvest them, we can do a lot of molecular biology on these mice. So we use biomarkers, biologic aging clocks, and a variety of other phenotypic markers of aging in these mouse models. And these are this is actually old. This is some of the results we've seen with different interventions. I should thank Jacinda, who's hanging out here somewhere, for really organizing these studies, and a lot of other people in the lab that support them. One of the things you'll find is that these are interventions that either came from our lab or mostly came from other labs. We can repeat about half of them, so that's not too bad for reproducibility and longevity interventions. But you'll also notice that there are things that only work in males or only work in females in our hands. And that's reflecting the fact that there are huge sex-specific effects on aging here. We're starting to see that in the human studies as well. And I think that reflects the fact that sex hormones are driving aging. And in humans, when you add menopause on top of that, that's a field we really need to focus more on. I want to talk about this guy just briefly. He came up to me about, I guess it's 4 months ago now, and pardon my French here, but he said, "Oh, you're that doctor that talks shit about me." I said, "I'm not the only one." Actually, he was a very nice guy. We talked for about 40 minutes, but I think that I have an issue with how he promotes the longevity field, and that's because of combining interventions. He thinks that if you have 20 things that work, you can just stack 20 things together and you'll get 20 times the effect on longevity. And there is zero evidence for this. And we've done a lot of mouse data combining different interventions. The most common thing you get is that they cancel each other out, not that they're additive together. And so I support the idea of longevity hackers. If they want to learn and do whatever they want to themselves to try to live longer, I feel like that's their right. But I also think they should be responsible for marketing good science. And in this case, it's not, because nobody knows how to combine different interventions together and how to make that synergistic effect. So anyway, I won't get too detailed on that. I want to talk about a drug we identified many years ago, though, called gemfibrozil. Actually, it was identified by Park Davis in the 1970s. We linked it to longevity. This is the original data from Park Davis, where they showed that when you give this drug, it lowers triglycerides and it increases HDL. And that's led to gemfibrozil being one of a class of drugs called fibrates that are still widely used for hypertriglyceridemia. We found that it extends lifespan using the non-vertebrate models first, yeast and C. elegans, And we've more recently shown that it has a pretty significant reduction in frailty in mice. And so this is an interesting drug because it has a very good safety profile. It's already clinically approved. It's off patent, although 6 or 7 companies still sell versions of it today. The confusing part came when we started looking at mechanism. So Chong He was a postdoc in my lab back at the Buck. So this project's been going on a while. And Charmaine, who's also hiding somewhere in the audience, is a postdoc working on the project now. Now, this drug is, like I said, is a class of drugs called fibrates, and these drugs are supposed to go and bind to this transcription factor called PPAR alpha and keep that transcription factor in the cytoplasm. So it blocks the activity of PPAR. Oh, sorry, I'm saying it backwards. Forgetting my science these days. It binds to PPAR alpha in the cytoplasm and dissociates it from a repressor complex, allowing PPAR alpha to go into the nucleus and activate genes involved in lipid metabolism, and that helps reduce triglyceride levels. There are other fibrates like clofibrate and others that are used on the market to do this, and they probably do have this activity. What I'm here to tell you, though, is this mechanism is not correct. It's not correct for longevity, and it's also not correct for reducing hypertriglyceridemia, I believe. And so I'm going to show you what the drug is doing today. And to do that, we've collaborated with Paul Nordland and people at A-Star to look at SETSA analysis to try to identify binding partners for the drug. And this is something I think we need to do more of, especially in longevity. We have a lot of drugs and small molecules that it's slow aging. But for the most part, we know some of the downstream hallmarks and pathways that they hit, but we don't know a lot of their binding partners. And until we know exactly what these drugs are binding to, it's going to be hard to combine them together. So this is based on a thermal shift assay. So when a drug binds to a protein, it tends to stabilize it at higher temperatures. So you can incubate an extract of cells with plus and minus the drug and look at proteins that are stabilized. And so we did that, and one of the big hits out of that was this drug, this drug PepT1. And you can see that on the Western blot on the top, at higher temperatures, the drug is stabilizing PepT1. And so we think this is the binding partner that's important for the longevity phenotype. And now I can't advance. There we go. So what is PepT1? It was identified in 1994. It's a amino acid transporter that's present in enterocytes in the intestine. Now this is, everybody that took biochemistry thinks about specific amino acid transporters like a tryptophan transporter. But most of your amino acids in the gut are taken up by receptors like this that are non-specific. They take up dipeptides and tripeptides, and they account for about 70% of the amino acid absorption that you get from your diet. And so this drug, we think, is binding to this protein and blocking amino acid uptake. Now, that kind of makes sense because we know amino acid restriction extends lifespan just like calorie restriction does in animal models. And a lot of people are on this kick right now where they eat lots of amino acids and fat and no sugar, which may be good for your short-term health, but in animal models, that's not the way to get to longer lifespan. It's by cutting calories. And actually, an isocaloric situation is a high-carb, low-protein that's associated with longer lifespan, not the reverse. Notice I said isocaloric. Carbs are the easiest way to get to too many calories, and in the mice we're giving complex, healthy, relatively healthy carbs rather than simple sugars. But anyway, reducing amino acids can extend lifespan. And so we're trying to get funding to develop this drug, and we can't easily do that by doing longevity because we need biotech investors. And so we've chosen a disease to go after. And so we wanted to look at first of all, can we confirm that this drug is actually reducing amino acid uptake in the gut? And then I'll tell you what disease we focused on. So this is a 2-week experiment where you give a labeled dipeptide, fluorescent, and then you can harvest the intestine and measure the uptake of that fluorescent dipeptide across the intestinal wall. And what you see here is that if you look at the bottom, when you have gymfibrozil, you can reduce the levels of essential amino acid in skeletal muscle by about 10% doing this. So the uptake experiments are on the top and the levels in muscle are on the bottom. Now, a 10% reduction in essential amino acids is, I think, a pretty robust effect. Obviously, if you reduce amino acids too much, you're going to have detrimental effects on the animal. But these animals are eating high calorie or high protein diet, high energy diet. And so reducing their amino acids is likely to have a beneficial effect. And we know that one of the best ways to reduce mTOR signaling is by reducing amino acids, because amino acids are the signal that drives mTOR activation. And that's exactly what we see when you take cells in culture and you use that fluorescent dipeptide to look at cell uptake. If you add single amino acids, which are taken up by different transporters, gemfibrozil in blue doesn't block amino acid uptake. But if you add a dipeptide mix on the far right, where the PEPT1 is required for uptake, then gemfibrozil reduces mTOR signaling. So we think this is another way of reducing signaling in the mTOR pathway in the gut and maybe in other cells in the body. And that's probably accounting for the longevity effects. So now to the disease. We were thinking about what we should try to do, and Charmaine had the idea to look at inflammatory bowel disease. And the reason for that is that this transporter takes up bacterial dipeptides and tripeptides, and that's part of the inflammatory response you get when you trigger when you have a triggering event in inflammatory bowel disease. And so if you can block uptake of those bacterial peptides, you can reduce the effects of IBD. And this pathway is relatively novel for this disease. The drugs that are out there are targeting different pathways. So we used a model of induced colitis in mice, where you, for 12 days, give dextran sulfate to the mice. And that causes a huge inflammatory response in the intestine, and we tested whether gemfibrozil could block that effect. I should say this is a collaboration with Zhang Yongliang's lab here at NUS. And I want to get back to aging, but I'll show you this slide. I think the key thing is on the bottom here with the immunohistochemistry. You can see with the DSS-treated mice on the left, there's a huge inflammatory response in the colon, and that's almost completely suppressed with gemfibrozil-treated mice. So we think this, I can't see that far, sorry. So we think this is a very interesting developmental project, and the idea now is to derivatize gemfibrozil to make new chemical entities that better hit this dipeptide transporter. We also know that the proteins that are important for the hypertriglyceridemia phenotype, and we're looking into that as well to try to improve the drug. So this is an example where you can start with a longevity pathway and get to a disease indication and hopefully take a biotech developmental route. I just want to come back a little bit to aging in Singapore. Another option you can really look at trying to understand data from is suboptimal data. And one example of that is this cohort from Trumi Labs that Camille is going to talk about later today, so I'm not going to talk about it. This is data that a company that's doing a biologic aging test collected and looked at the effects of different supplements on biologic age, and he'll talk about that data, so I'm going to skip that. And then there's also local cohort data, and so we're a number of groups now are looking at biologic age across a wide range of cohorts in Singapore. I want to show you just a little bit of data to give you a flavor for what's happening in Singapore. And so we had access with collaborators to the SG10K cohort. So a number of years ago, Singapore decided that they needed to do genome sequencing at a large level. So if you look at the ethnicity in Singapore, it's 74% Chinese and then Malay and Indian. So together, those three ethnicities represent a large number of people on Earth, but they only represented 11% of the genomes in databases. And so there was a need to expand that and look at more genomes of these ethnicities. And so they started with this SG10K project with restricted demographics, molecular phenotypes, some metabolic markers, and then full genome sequencing to understand what's going on. And so this project is a collaboration with Nirja and Joanne there at the top. And the idea was to look at biologic age because they did DNA methylation sequencing, or they did arrays in this 10,000 people. Now, the SG10K was sort of a compilation of cohorts that were available. And now they have completed, I believe, the sequencing of 100,000 people in Singapore. And the ultimate goal is to get to 1 million or maybe beyond so that we have a large portion of the Singapore population sequenced. But I'm only showing you data on the SG10K. So here are the 6 cohorts on the bottom and 4 biologic aging clocks on the different bar graphs. And the general conclusion from this slide is similar to other studies that have done this. The clocks don't all perfectly correlate with each other. That's been seen with biologic aging clocks in a wide array of studies, although there is some correlation. The clocks don't work perfectly in these Asian populations, but they do work pretty well. So that's encouraging. And so we can draw a few conclusions, and actually there's a lot more data on this I'm not showing you. It's all still unpublished. One thing is the Chinese ethnicity is biologically younger than the Malay and Indian ethnicities. That could be socioeconomic status. It often correlates with socioeconomic status in other parts of the world. On the right, you can see that suboptimal metabolic health is associated with higher biologic age. That's not surprising. It's seen other places. The middle result is quite interesting, which is that we can already see that baby boys are older than baby girls at birth. This is from Yap Singh and his colleagues' Gusto cohort. That's fascinating. It suggests something's happening in the womb that's already influenced the aging process and reinforces the notion that aging doesn't begin at 60 or 70 or 40. It begins maybe even before birth. And I was just talking about a collaboration with another group where we're going to try to look in great detail about what's happening at biologic age very early in life. Maybe there's a genetic conflict with a male genome and a female body. We're still unsure why this is affecting, but I think it's fascinating that we can already see this sex difference. There was genetic variance associated with epigenetic age variation. I won't go into it for the sake of time. Some interesting genes come out, PARP, Wnt3A, some things already linked to aging, and we're trying to look into that in more detail. So let me just conclude with a couple of slides. You know, what's really needed to accelerate this healthy longevity in Singapore and actually globally as well. And this is something I have to give credit to our dean, Chong Yup Singh, because he helped with these slides. So we need outcome measures that are validated. Most of the biologic aging clocks are effective at the population level, like the data I just showed you, but they're still not validated at the personal level. But we need to know for an individual whether they're slowing their biologic age or getting biologically younger from an intervention. And we have to have measurements that can do that effectively. Ultimately, we need precision. We need to know which people are going to respond to which interventions, and that's still a work in process. These clocks have to be responsive, and I want to reinforce that. Jan Gruber is going to talk about this tomorrow. We also need to test a wide array of interventions that may slow aging. They noted this intervention testing program on the right, which tests mouse aging. They test about 5 or 6 interventions a year. It's been going for like 20 years now, and published the first rapamycin paper in mice. But we need to do that in humans now. We need to do a wide array of interventions in middle-aged or older people, looking at which ones affect biologic age in which populations. And then also we need to figure out how to deal with regulation in this space. Longevity clinics are doing lots of different things. You can go to Honduras, and I'm on my last slide or so, don't worry. You can go to Honduras and get gene therapy for aging now if you want to spend $40,000 and you're interested in putting telomerase in your body. I don't recommend that, but my point is that in various parts of different regulatory environments, you can do a lot of different things now to try to slow your age. You could just say don't do any of that, and actually I don't think Singapore should do gene therapy, but I think that's also not a way to go forward. These are healthy people, mostly trying to make choices to slow their aging process. And I think outright banning it is just going to drive it underground. It's already a wide market. I would estimate 50 to 100,000 people are taking rapamycin right now to slow aging around the world. And there's a lot of repurposed drugs, nutraceuticals, and devices that are available, some of which I think are reasonable and we should be testing, and many of them I think were reasonable and we should be testing. So one way to do it is to have a different kind of regulation where you have more tolerance to interventions in clinics, but also force the clinics to share data behind a government-controlled secure repository so that they can validate the safety of the particular interventions that are being tested in the clinic, look at the ethics, and then also if things work with those clinics to expand it and get it to the entire population quicker. So already places like the Bahamas are trying to do that. Montana in the United States has done something similar to that. I think it was a great opportunity for Singapore to try to take this on and do things that are safe and efficacious and become a center for sort of longevity science. So that's something we're trying to get some traction on. So that's all. These are just people in my lab that have helped out. I think I mentioned most of them along the way. I didn't talk much about data today. Sorry I ran over, and thanks for your time.
Thanks, Brian, for the nice talk. We have time for a couple of questions.
Stunned silence. Yeah.
Yes, that is.
Thank you for the great talk. As you mentioned, like the biomarker of the aging, and then, but recently many of the teams published the different biomarkers in terms of the. Sometimes people use O-link or SomaScan, which is the peptide. So in the future, I believe the like some the phenotype associated the biomarker is very necessary, not just the predict age. So what type of the biomarker is?
Well, first of all, I don't think there's anything special about methylation. The biologic aging clocks were originally derived around methylation measurements, and most of them are methylation today. But I think you can take any complex data set with enough people and enough data and generate a biologic aging clock. I don't think there's anything primal about looking at DNA methylation. I think the short answer is that different clocks are more useful for different purposes. So I agree. I think the proteomic clocks and the lipidomic clocks and the metabolomic clocks are really exciting if you want to understand mechanisms of aging. Because if you find 20 methylation sites that predict age, then you don't really know what those methylation sites are doing. If you find 20 proteins that predict age, you have a pretty good idea maybe what those proteins are doing and what that means for the aging process. I also think that there are different generations of clocks. There are first generation clocks that predict your chronologic age, and second generation clocks that predict outcomes, like the one Jan Gruber will talk about tomorrow, which is based on clinical markers. And then there's clocks that predict pace of aging and organ aging and other things. So I don't think we're going to find one clock that answers every question. We have to use the right clock for the right purpose. In the clinic, what we need are actionable clocks. We need clocks that you can look at and you can say, we need to go in this direction to try to influence your biologic age or mortality risk. And so, we've been heavily focused on trying to develop those because we want things that can make a difference in clinics and hospitals. So I don't think there's an easy answer to that question. But you also have to consider cost. If you're going to expand things, you can't spend $5,000 measuring somebody's age. You have to spend $100 or $200. So all of these factors come into play, and I think that trying to, first of all, when you can use many different kinds of clocks, and if you have to choose based on what you're trying to achieve.
Thank you.
Okay, we have one more question.
Thank you. Thank you for the presentation again. A question, because you're out there exposing the science, and maybe one conflict you have with Brian Johnson or the people that I also have, but is nowadays how we should improve our communication, because here, where everybody's doing science, we try to improve health, but unfortunately what we see is a lot of people without knowledge or doing N of 1 experiment, they receive a lot of attention. Every time you go, if there's a, again, I'm not against biohackers, but if they're doing something, everybody's interested, oh no, you should do this peptide, you should apply this, and how we should improve in this part.
Yeah, first of all, I don't think you're trying to say this, but let me just say, I don't think you have to be a scientist to talk about aging and longevity. But yeah, I know. I think that if you're really trying to make a difference, though, you have to have enough understanding of the science to make things that are scientifically based and really trying to help the population. And this is really the Wild West right now in the longevity space. You have influencers, you have people like Peter Attia. He's a doctor, and he's like, I may not agree with everything he says, but he's highly knowledgeable in the space. You have people like Aubrey de Grey, who I would have said was not particularly knowledgeable at the start, but now he's very knowledgeable in the space. And so there's a wide array, and I think it's difficult right now for people in the public to know who to believe and who not to believe. And that's one of the reasons I've spent a lot of time doing talks to the public these days, because I'm trying to help in that regard. But it also reflects the interest in people that want to live longer, be healthy longer. So I think this whole movement is generally a good thing, but there's a lot of buyer beware out there at the same time, and it's hard to manage it for sure.
Yes, very nice talk, Ryan. So I have a very small, curious question for you scientifically. On your GEM fibrosil, when you use it for treating IBD, have you compared the results that you get with your compound and more standard ways of treating IBD, for example, corticoid treatments, to see whether there's an improvement there?
We haven't tried it against standard of care yet. That's the next step in mice. And in addition, it might be additive with standard of care because it's a different mechanism. So that's something that's on the agenda to do, and we just don't have those results yet. It's a good question.
Thanks.
Thank you, Brian.
Thank you.